Hi everyone, and thanks for joining for another Inspire series by Acchutha Institute of Optometry. Today we have Dr. Shrikant Bharadwaj, who doesn't require any formal introduction. He's a well-known person in optometry and vision science field. So to quickly introduce himself, he did his bachelor's in optometry from Elite School of Optometry, and then went on pursuing his PhD from School of Optometry, Berkeley School of Optometry in US. Current and he came back to India in 2009 or 10 and joined the Prasad IA Institute, and currently he holds the position of senior scientist at the Professor Brainfold and Eye Research Center, and he is also a Fulbright Nehru Academic and Professional Excellence Fellow. And today he is going to share about what can optometrists learn from engineers and mathematicians when solving clinically relevant problems with respect to keratoconus and the experiences from his research lab. And thanks again for joining Dr. Srikanth. Over to you.
Thank you so much, Gopi and Sharanya. This is a real pleasure for me to be part of this meeting. I completely appreciate this invitation. Let me make sure my slides are visible and I am audible also, I'm hoping, right?
Yes.
Okay, great. So, good afternoon, everybody, and I know this is a middle of the work week, and thank you so much for taking time to come and attend this session. The topic, as Gopi was pointing out, is what can optometrists learn from engineers and mathematicians when solving clinically relevant problems? And I'm going to share some lessons that I have personally learned in pursuing my research on keratoconus for the past a decade or so in this area. So the key words in this topic are optometrists. I mean, I'm assuming majority of us sitting here and listening to this talk are optometrists by training. And engineers and mathematicians, these are the other two professions that I have learned quite a bit from, or I use a lot of techniques developed by engineers and mathematicians. And the other one is a clinically relevant problem. And I think this is where the link between how a medical field can actually work with some of the basic sciences can actually work out quite well. Like I said earlier, I'm going to share some of the lessons from my work on keratoconus. So as we move along in this topic, what I'm hoping to point out to you is it's a worthwhile endeavor for us as clinicians to interact with engineers, mathematicians, and essentially people of other domains. They bring in a lot of relevant information that we can use all towards solving the patient's problems in the end. So before I start, I can give you a broad overview of what my lab does. So I am the principal investigator for the visual optics and psychophysics laboratory. By the way, Gopi, can you confirm if my mouse is visible? The little cursor is visible. It is? Okay, great. Thank you. So I am the principal investigator for the Visual Optics and Psychophysics Laboratory. And for those of you who are interested in the lab profile and less so in my profile, you are welcome to scan the QR code and then it will take you to the lab website where you can learn more about what we do. But very broadly speaking, we do work in 3 or 4 sort of related areas. The broad question that we ask in the lab is if the eye is a light capturing receptacle, it captures photons of light and it forms information on the retina. That information is essentially decoded by the brain, and that it's the decoded information that allows us to see the world the way we see it. In its full glory, all the fine details in color and motion in 3D space and all of that. So when there is a front end limitation in the eye, what sort of impact does it create on the way we reconstruct the image in the brain? Oftentimes, this front end limitation need not be the same in both eyes. It can be one eye can be blurred, the other eye can be clear. So under those situations, when does the brain choose to combine information from the two eyes? When does it choose to say, no, I don't want to deal with the blurred information. I am going to go ahead and ignore that input. What sort of rules govern this? What sort of computations goes on? Are sort of the broad areas of research. Within this ambit, we look a lot about the near vision complex, studying accommodation, vergence, pupil responses and stuff. We also study disease conditions like keratoconus, which is going to be the major focus of my topic today. Recently, we have also ventured into the field of visuomotor performance, and I'll put that in the context of keratoconus in just a bit. And there's a small component of technology development that I am also involved in, specifically in the development of objective pupil measuring devices. So we'll primarily use keratoconus as a model and try and look at how that broad question of the front end limitations impacting perception happens. In that process, in order to solve that kind of a question, we have been working with the people from many other disciplines using tools developed in many other disciplines, which is what I'm going to sort of focus on and give you some of my learnings and some insights as we move along in this journey. So acknowledgements are due to a whole range of people who have contributed to the overall work in the lab. The present lab members and the immediate past lab members are shown in the picture here. Very, very fine folks who have helped me run the lab. It is their blood and sweat that I am representing here. I should also acknowledge the various funding agencies that have supported my research over a period of time. Both the government and non-governmental agencies, industry that has supported this work. Several collaborators for the work that I am presenting. Some of you may recognize Prem Nandini in this picture. So for the purposes of this particular talk, I am going to call these three individuals as clinicians. The ones that are in the red box are all optical engineers and bioengineers of various disciplines who have helped work with me on various areas. And these three individuals here have an experimental psychology or a mathematics background from whom I have learned tremendously for solving some questions that I'm going to present to you today. So right away, I want to set up the stage that the work that I am presenting is not going to be purely clinical. The work that I'm going to present involves a lot of engineering principles, primarily optical engineering principles and experimental psychology techniques. So I will try and present this in a manner that is palatable to all of you. But if there are any questions, feel free to put it in the chat box, and I'll be very happy to talk to you about it. Specific to this work, acknowledgements are due to all of my students who have contributed to the body of work that I'm going to be presenting today in one form or the other, and I will point out to their work as we move along here. All right. So thanks to Gopi, which I thought was fantastic, because he ended up collating a set of questions that the registrants have already asked. And this, I think, is remarkable because it really helped me set the tone for the presentation. So I'm going to broadly divide these questions into two or three parts. These are some generic questions that came in the registration desk. I will cover them as we move along in this topic. There are some specific questions related to computer simulation, geometric modeling, wavefront aberrations, impact of contact lenses on wavefront aberrations in keratoconus. This will be covered in the first two parts of my talk. And there were a lot of questions related to AI modeling, artificial intelligence, machine learning and stuff. And fortunately, I have worked with some people to develop these models. So I will be in a position to answer some of these questions in the last part of my talk. So one way or the other, most of these questions are going to be answered. There are some very generic questions as to, why am I doing this research? What inspired me to do this? I'm going to sort of park those for a minute, and I will be happy to chat with you offline for answering those sorts of questions. All right. So the fundamental premise of my talk is shown in this diagram. This is my version of Pablo Picasso's diagram, very famous diagram that my students keep seeing this all the time. I know some of them are in the audience, so my apologies for going through this rigmarole once more. But fundamentally, I think this diagram is very powerful because it tells us about what we see and what sort of limits the kind of vision that we are experiencing. So what we see in the external world is represented in the form of this eye chart. This could be any other natural scene that you're seeing. Light in the form of photons is going to get into the eye, gets refracted through the cornea, crystalline lens, and all of that, and forms an image on the retina, which converts this into electrical signals, sends them through the optic nerve, LGN, all of that, and you reach the visual cortex where the scene reconstruction starts happening. The scene reconstruction is then sent off to a dorsal stream and a ventral stream for perception and action related activities. In this scheme of affairs, what is there in the external world in front of us, once it goes through the eye and the brain, there is a certain amount of filtering that happens in that information. So the first level of filter is the optical filter. There are limitations that are imposed by the eye itself that limits how much of details can we see. For instance, you can't see the coronavirus that plagued us a few years ago because it is too small in size, too small for the eye to resolve those fine details. So our retina, our cornea, and optics impose a certain limitations on how much we can see optically. If you surpass that filter, you hit the neural filter, which is the brain, and the brain further filters that information, and it only gives us a low pass filtered version of whatever is there in the external world. So what we see of the external world is only a very sparse representation of the reality that is out there. So between these two filters, there is certain things that we end up seeing. There are many things that we end up not seeing under physiological conditions. If you now add a pathology to it, and keratoconus being a pathology primarily of the optics of the eye, there is additional layers of filtering that happens to this image. And the brain now has to deal with this filtered image and be able to make sense of whatever distorted image that is being sent for processing. So what is the nature of this filter? What sort of distortions happen? How does that limit my perception is essentially going to be the crux of this talk. Another way of thinking about this is in the form of a structure function relationship. If you think of the eye as an optical structure that is meant to subserve functions, your cornea, your crystalline lens, your retina are all anatomical structures that allows me to form an image on the form a representation of the image. Now using that image, we do certain functions, we do certain motor actions and things like that. So how much of the structure is going to define how much of the function that we do is another way of thinking about it. When the structure gets degraded, is there a commensurate loss of function? Is there a commensurate loss of functional vision? And eventually, does that impact the quality of life of an individual? So this is the sequence that we are actually going to go through. So in the context of keratoconus, we know that the disease is a bilateral corneal ectasia, oftentimes asymmetric in severity. It's progressive. It affects people in the early teenage and tends to sort of continue to impact vision of these individuals. Cornea continues to thin and get steepened. Eventually, it will lead to scarring, and you may need to now go ahead and replace the cornea. But fortunately for us, we now know once you detect keratoconus, we have ways to fix it. We have collagen cross-linking kinds of procedures that strengthen the cornea and prevent or at least reduces the aggression with which the disease is progressing. We also know how to optically correct it using a variety of contact lenses that improves the vision of these individuals. But fundamentally, from that structure function relationship point of view that I was talking about, what you are now dealing with is an optical structure loss in the cornea. And this distorted corneal structure creates distortions in the retinal image quality. It degrades the retinal image quality. And the degradation in the retinal image quality impacts visual function. And because of an impaired visual function, you now have poor functional vision. I am differentiating visual function from functional vision here. Visual functions is primarily what we end up perceiving, your standard acuity, contrast, stereo sorts of things. Functional vision is using that visual functions. How do we interact with the external environment? How do I navigate? How do I move my hand to go grab something in front of me? How do I give high five to my friends? So on and so forth. So the source of all these problems is the front end limitation in the eye. That leads to a cascade of degradations that we are now having to deal with. And the talk is going to essentially progress along these lines. So we will spend quite some time trying to understand what is the limitation in the retinal image quality. We will then talk about what sort of limitations we find in visual functions and our more recent explorations on some functional vision limitations that we have found. And we'll sort of tie all of this together in the context of the structure function relationship. Okay. All right. So in this context, from the title of the talk, where have I involved engineers and mathematicians? For me to understand the optical quality of the eye, in order for me to understand how does it impact visual functions, I have had to use a lot of optical engineering techniques. Wavefront aberrations, reconstructing the retinal image quality from the wavefronts. All of these are very fundamental Fourier optics techniques, and I have learned all of that from optical engineers and electrical engineers. We owe these fields quite a bit for the kind of contributions that they have made to our understanding of vision. Now, when it comes to functional vision, a lot of techniques have been involving experimental psychology techniques, psychophysical techniques that helps us talk about behavior of individuals, that helps us think about how does a person respond to a certain stimulus that they have been presented with. So here I have gone back to mathematicians and the techniques that they have developed, applied that to disease conditions like keratoconus and others that we are going to talk about. So built into whatever I'm talking about is a huge influence of engineers and psychologists and mathematicians in the research that we have been doing. So current diagnostic standards for keratoconus essentially involves one way or the other scanning the cornea and getting an estimate of the surface properties of the cornea, either in the form of a topography or in the form of a tomography of the cornea. You get some surface characteristics of the cornea from which you now derive a whole load of different metrics to say how is this surface distorted. Turns out there are close to about 70-80 metrics that are there that you can that some of them that are built into devices like the pentacam, from which you can estimate, okay, how much is the cornea getting distorted, how much is it continuing to distort because the disease is progressing, so on and so forth. The one metric that is currently gaining a lot of popularity, and we have been using that technique, you have been using that metric, sorry, is the thing called as a D index. The D index is a multi-metric index in the sense that it takes into consideration the curvature of the cornea, both anterior and posterior curvatures. It also takes into consideration the thickness of the cornea. So it's based on almost the entire cornea's profile, and using that we have been able to grade the severity of keratoconus. So in the slides that you will see moving forward, you will see where we have plotted the visual functions as vision as a function of disease severity, and all these disease severity is using the units of the D index. This is in lieu of some of the older techniques that we have, older metrics like the Amsler-Krumich classification that we have used, which is primarily based only on the keratometry values. The D index have been shown to be more superior in terms of detecting and detecting the progression of the disease condition. So we know that the cornea gets progressively distorted in keratoconus. The more distorted it is, higher the D index is, more severe the keratoconus. That's the bottom line in all of this. Vision loss is a little bit more complex. What is plotted here is work primarily done by Preetham Kumar in my lab. He was a PhD student. Currently, he is a postdoc at University of Houston College of Optometry. Preetham did this as a sort of a hybrid longitudinal study where he followed up patients with varying severities of keratoconus and plotted their visual acuity, contrast sensitivity, and stereo acuity. The solid line, notice that I am using these terms of normalized visual acuity and normalized contrast sensitivity. These are normalized to the data of controls, age-matched controls. So if somebody has a visual acuity of 1, which means their value is equal to those of the age-matched control. So anything lower than that is progressive worsening of vision in these patients. You can clearly see that visual acuity and contrast sensitivity has a monotonic loss with an increase in disease severity. The more severe the disease gets, there is a progressive loss in these functions. Acuity seems to show some kind of a flattening initially and then it drops off steeply, but contrast sensitivity right away starts degrading, worse and worse as the disease progresses. Stereo acuity, on the other hand, seems to be worse from the get-go. No matter what your disease severity is or how different the disease severities in the two eyes are, stereo acuity seems to be degraded in keratoconus. So poor stereo acuity is something, is an early hallmark feature that the two eyes are getting distorted images and the distortions may not be similar in the two eyes. I am emphasizing on this because at least in routine clinical practice, if you think of keratoconus primarily as a monocular disease condition, you focus only on the monocular acuity and monocular contrast, if at all. But the underlying binocularity of the individual is already taking a major hit, even while your acuity and stuff may be more or less of comparable, more or less uncompromised yet. So question is, how do the vision loss explained by the underlying loss in the optics of the eye? What is the connection between the two? In order for me to understand this, I need to go back to my fundamentals of measuring the retinal image quality. And here is a schematic of how this is done. So take a simple case of light rays, parallel light rays entering into the eye, getting refracted at the level of the cornea and the crystalline lens and forming an image on the retina. What is shown here is an aberrated myopic eye where the image that is coming out, that is refracted, is not focused on a single plane on the retina. It is sort of distributed across multiple planes within the retina. Now that is happening because of the cornea. In the case of keratoconus specifically, the cornea is not a uniform refracting surface. It has now gotten steep at certain areas, flatter at certain other areas. So light rays entering through the cornea is not getting uniformly refracted. It's getting refracted by different amounts and it's forming a multifocal image on the retina. So effectively, a keratoconic eye can be thought of as a multifocal optics, a multifocal optical system. The consequence of that is the point spread function on the retina, which is how distorted a point of light can get on the retina, is extremely complex. It is not a simple myopic, defocus, not a simple astigmatic defocus, but a very complex form that you need to break it down into the component Zernike polynomials. Now, this is an optical engineer's bread and butter. They do this on a day-to-day basis where any wavefront, we deal with eyes wavefronts, but they deal with wavefronts coming from atmospheric turbulence, they deal with wavefronts of radio frequencies. A whole was describing that the pattern of blur that is formed on the retina is a very complex pattern and that needs to be broken down into component Zernike polynomials in order for us to understand the pattern of blur that you're experiencing. Now, from the Zernike polynomials, I can again use fundamental Fourier optics techniques to reconstruct what will be the nature of that blur. How do I quantify that kind of blur on the retina? Turns out that our optical engineering friends have come up with close to about 31 different metrics in the way I can define the quality of the blur, define the amount of blur, define the quality of retinal image that's there. I can define the quality simply based on the wavefront data that I am directly getting from an aberrometer. I can also define it based on the point spread function, the complex point spread function that is represented here, or from the Fourier transformed version of that point spread function, which tells you something about the kind of spatial frequency information that is available in the retinal image. So using any of these three techniques, I should be able to say how blurred is my image on the retina. What is a very qualitative experience of the patient saying I am now seeing things blurred? I am now able to quantify that kind of an experience using these techniques. Now, when I do that for keratoconus, what do I end up finding? In the retinal image itself, you see that the wavefront aberrations of the eye is roughly about 5 to 6 times in keratoconus. Every eye, your eye, my eye, everybody's eye has a certain amount of wavefront aberrations. And this is typically about 0.3 microns for a 5 mm pupil size. So none of us are free from these wavefront aberrations, but compare the values of what you get in a control to what you get in a keratoconic eye. If ours is about 0.3 microns, they are hovering around about 1.5 to about 2 microns, close to about 5 times, 6 times the amount of wavefront aberrations. Notice also one other interesting thing. Between the two eyes, our retinal image is very, very similar. The interocular difference in the wavefront aberrations is nearly zero. Whereas in a keratoconic eye, high amounts of interocular differences is there. Now, what this means is the quality of the retinal image formed in the left eye and the right eye are very different from each other. And that's going to challenge the process of vision a lot more than having similar qualities of image in the two eyes. Once again, I'm trying to drive home the point that think of the keratoconus as a binocular problem where the patient may be experiencing all kinds of difficulties with depth perception. If I use that Fourier optics techniques and I reconstruct that retinal image, what would a keratoconic patient be seeing? Here is some of us with some amount of higher order aberrations, and notice what a keratoconic patient is seeing. There are two obvious things that goes on in this retinal image. One is the overall loss of contrast. This image appears duller compared to a template eye chart without any kind of blur. The second thing that is very obvious in this is the doubling or the ghosting of the letters. A typical keratoconic keratoconic patient will complain of that, saying, well, I'm seeing a ghosting of the same letter behind whatever I am, whatever that letter is. And this ghosting or doubling can actually cause a lot of problems for these keratoconic eyes. And it's arising from the underlying pattern of wavefront aberrations. Now, it turns out both the contrast and the ghosting have a negative impact on the way on the way the keratoconic patient is seeing. So both these studies essentially quantified the amount of loss that they can experience. One of the ways optical engineers actually do this is by constructing something called as a through focus curve. And what I'm plotting is a through focus curve for a control. I'm going to quickly explain this and get to the values of keratoconics. So the through focus curve is essentially saying if I now have an object that is coming from beyond optical infinity, essentially these plus values here, through optical infinity 0 and to closer viewing distances, what sort of quality of image that I'm going to experience. If I do not have ocular accommodation, if I am considering the eye as a monofocal single focus system, what will be the quality of the image that I am getting? If I am an emmetropic eye, I get the best possible optical quality close to 0 diopters at optical infinity. Anything closer, uncorrected near vision, and uncorrected vision beyond optical infinity, both of those are going to be blurred out for me. So there is a systematic drop in the retinal image quality as I am getting closer than optical infinity in these patients. So a very characteristic of a monofocal optical system. Compare that to a keratoconic eye. A keratoconic eye's through focus curve is very flat. There is hardly any change in the quality of the optics across a whole range of viewing distances. The peak image quality, that was a very distinct peak that you could see for the controls, is completely absent in keratoconus. They are sort of having poor image quality, and that compromised image quality now exists over a whole range of viewing distances. Once I put on an RGP contact lens on this patient, there is a little improvement in the quality of the through focus curve, but it never sort of reaches the level of a controlled population. What's the consequence of this? No matter what kind of blur that you give your keratoconic patient, the vision in them is going to be more or less the same. That's what this through focus curve is telling you. So when I am trying to do a subjective refraction on these patients, when I'm trying to create the end point of subjective refraction, you're going to have a hell of a time trying to refract a keratoconic patient, because no matter what lens that I am going to put in front of this patient, they are going to say that my vision is more or less the same. So large variability in the end point of subjective refraction is something very obvious in keratoconus, and as clinicians, you guys will know this equally well, that the refraction end point of subjective refraction can be variable by almost 2.5 diopters in a keratoconic eye across a whole range of axes of astigmatism. All of that is arising because of the underlying poor retinal image quality, is what you're saying. So before you blame your refraction skills or your students' refraction skills while you are dealing with keratoconus, bear in mind that it may very well be the optics of that patient that is making them give a lot of variable responses. So this is one thing that we need to recognize when we are managing these patients, and that information really has come from the optical engineering techniques that I was talking about. Now, coming to binocularity, notice that I made a point earlier that the quality of the retinal image between the left and the right eye can actually be very different in keratoconus. Now, as a result of that, if the control eye is now seeing similar qualities of point spread functions in the left and the right, you actually are able to get good quality correspondence matching. You are able to match the left image and the right image very nicely in order to get a good quality retinal disparity signal. Now, this is a mathematical technique called as cross correlation. So, a cross correlation may you essentially match whatever points in the left eye is there to whatever point in the right eye. That tells us how does the brain actually put the two information together. And a good quality cross correlation match is what you are seeing in a controlled eye. Whereas when you have a keratoconic patient with all kinds of different qualities of image in the two eyes, the cross correlation signal is becoming poorer. The keratoconic eye is not able to match the information from the left eye and the right eye properly, leading to impaired retinal disparity processing. Now, fundamentals of binocular vision should tell you that if retinal disparity processing is impaired, stereo acuity is going to be impaired because they are not going to be able to get the depth impression properly. Not only that is the problem. In addition to that, the issue is that we found the poorer eye in keratoconus may actually be suppressed. There could be increasing levels of suppression as we were measuring this using this parameter called the contrast balance point. I won't be able to get into the details of this, but this constituted the PhD work of my other student, Bhagya Marella, who is now a postdoc in Indiana University. So what she did was she measured how much is the weaker eye in keratoconus actually suppressed, and turns out that larger the difference in image quality between the two, deeper is actually the suppression in these keratoconic eyes. So not only is disparity processing impaired, the eye may also be suppressed, resulting in overall poor stereo acuity in these individuals. So what this is pointing out to us overall is that even though keratoconus may be primarily an optical pathology, there may actually be neural limitations also that could exist. So some form of, remember that keratoconus is a progressive disease, it's a chronic disease that continues to exist over a period of time. Once a patient is keratoconic, they are going to remain keratoconic for the rest of their lives. So the compromised quality of images in the two eyes can actually lead to some form of amblyopia in these patients, especially some form of meridional amblyopia in these patients is what you are concerned about. So keep that in mind again when you are managing your patient with keratoconus. Not only should the acuity in each eye be good, but the acuity should also be similar in the two eyes. That will support binocularity in these patients. All right, so this is what I was talking about. Now, when it comes to managing these patients with with contact lenses, there's a whole range of options that is available. Preetham did this work again, where he very painstakingly, fit 4 different kinds of contact lenses on these patients. Routine RGP lenses, Rosque, Kerasoft, and the large diameter scleral lenses. And he looked at what happens to these visual functions and retinal quality when it actually you're wearing these different lenses. The study design was a very complex one, so we had only about 25 patients or so in that sample, but that gave us a lot of insights in the data. So what I'm plotting here is logmar acuity, and as the severity of the keratoconus increases, the logmar acuity is essentially worsening. Now notice that when I put on a Kerasoft lens, there is an improvement in the visual acuity, but it never reaches the level of controls. The controls is the gray bar there. Any of these lenses, Rose K2 or RGPs or your fancy scleral lenses, they all improve vision more or less by the same amount. So between spectacles, compared to spectacles, certainly these RGP, certainly these specialty contact lenses fare better. But between these specialty contact lenses, is there a remarkable difference between the two? At least we were not able to find a big difference in the performance. Acuity, same story. Contrast sensitivity, it is the same story. Stereo acuity, also it is the same story. So between the different kinds of contact lenses, more or less the quality of vision in these keratoconic individuals are the same following the wear of these lenses. Optical image quality that was performed by Preeti, who is currently a PhD student in the lab. She found that the optical quality is exactly the same. You can compare the green, the red, and the brown traces. All of them are almost identical for these 3 lenses. Kerasoft certainly is faring worse compared to the 3. So the insight that we got from this is when you are offering solutions for your patient in the form of different contact lenses, the deciding factor for which lens you might want to pick for that patient will depend upon non-optical factors. When I say non-optical factors, it may be the fit of the lens in the eye, how well the lens is actually stabilized in a distorted cornea. It could be the wearing comfort of that patient, or it could be the cost of that patient. Visual functions wise, more or less all of them are almost identical, seems to be the story that's coming out of that. All right, so that brings us to the end of the first part of the talk, where we spoke a lot about retinal image quality and visual functions. And what we want to now move on is to some work on the functional vision. This is, as is indicated here, is relatively new work. This is not published yet. We have sent it out for peer review at this point, but I can, in fact, one work is published, the other is sent out for peer review. Now, for a minute, if you think about this, all of what we have been speaking so far, be it acuity, contrast sensitivity, stereo, all of what we are calling as threshold level performance. And what I mean by that is that's the smallest value that you can detect. That's the smallest size that you can detect, smallest disparity that you can detect, the lowest contrast that you can detect, and so on and so forth. Now look around yourself. When you look around this, most of our vision is actually not those smallest letters. Most of our vision is actually driven by larger letters, higher contrast letters, bigger disparities, what we call a supra-threshold performance. So if threshold level performance is this affected in keratoconus, what would happen to supra-threshold performance? In case you are wondering why I chose this particular picture of all the things, this was actually not chosen by me. This graduate student who is a Bengali wanted nostalgic feelings about the cars in Kolkata. So that's his picture. And you can, but this drives home the point very clearly that what you see here are very bright, high contrast kinds of targets. There are fine details, but what's going to define our perception for the most part are these high contrast targets. So what is the perception of individuals of keratoconus with high contrast targets? We used a very classic paradigm in experimental psychology called contrast constancy paradigm, where people have to match the perceived contrasts of two gratings, a low frequency and a high frequency grating. And they have to say, okay, are these contrasts the same or are they different? We figured that if the threshold performance is this poor in keratoconus, their supra-threshold performance should also be dramatically poor. They should not be able to see the world with the same amount of contrast that a normal person is able to see. But tell you what we found, the brain is beautifully adapted to that. The brain is able to completely adapt out all the limitations that you see in threshold level performance at a supra-threshold. So when a keratoconic individual is out there in the world, looking at large targets, broad, high contrast targets, they are not going to experience any kind of limitations in their performance. Any degree of keratoconus, it seems to be the same performance. In that paper, we in fact switched people from contact lenses to spectacles and immediately measured it. And the brain seems to be almost instantaneously able to recalibrate itself. So the brain is remarkably plastic and it is able to recalibrate itself beautifully for maintaining our supra-threshold performance. At a threshold level, we are all having all kinds of difficulties. So here is a case where you want to not discount the properties of the brain while you are dealing with a disease like keratoconus, but you want to think of the brain as a partner in crime, helping in some ways and hurting in the other cases. What about visuomotor performance? What we do in the real world are all of these real world activities. We drive in crazy traffic, we thread a needle, we pour water onto a jug, we play sports, we put a bulb in onto a holder, and all kinds of complex activities is what we perform. And every one of these activities actually requires a very keen sensation of depth. If the keratoconic individual is that poor in their depth perception, are they able to do activities like this? Are they limited in their activities is a question that we asked. And the additional question is, we measure an individual's stereo acuity using tests like this, your RAND test, your Titmus fly test, and all of that. And what we really do in the world are more complex activities like this. So anything in this measurement give us an indication of how they might actually be doing in those measurements. Does clinical assessment of stereo acuity match with the measurements seen in the external world? Question we asked, and Preeti, who is my PhD student currently, she took up this challenge and then she put up this super fancy contraption called as the buzzwire task. This task is a childhood game that many of you may have played. You have to pass a loop through a wire without actually touching it. If you touch it, then there is an error and you are actually penalized for that. Question is, when you have really blurred images like what I am actually showing here, are these keratoconic eyes able to do the task as well as a control eye or as well as a myopic eye that has uniform amounts of blur? Turns out that individuals in the keratoconics show some very interesting properties. Binocularly, we are all much better at this task. The green dots in this. I'm not going to get into the details of this figure. It's a complex figure, but I can tell you the summary of it. Binocularly, we are much better off at this task compared to monocular viewing. We make a lot more errors monocularly. But keratoconics have very limited binocular advantage. Their vision is poor. Because of that, their performance in the task is also very, very poor. Now, one interesting feature is when we are doing the task monocularly, we slow down. Let's say you're driving through fog and you are not able to see the scene well, you will slow down your car. That seems to be common sense behavior. Our keratoconic friends don't seem to do that. They seem to be going as fast, making a lot more errors. Now, is this because they are overconfident that they can do this task, or is it because they are, is it because they are overconfident, or is it because they are, is it a personality issue with these keratoconic eyes? I do not know at this point, though that seems to be a unanswered question in the literature. The pattern of blur also seems to matter. Myopic eyes with similar amounts of stereo acuity still seem to be doing better than these keratoconic eyes. So this is an area that we are currently exploring in this field. How does it compare to stereo acuity? Absolutely no correlation with stereo acuity. That's a disappointment, because whatever that we are measuring in the clinic as perception of depth does not seem to translate into these complex visuomotor activities. So we need to sort of watch out for what we are examining in these patients and what is the task that they are actually engaged in the real world. Just because their stereo acuity is good doesn't necessarily mean that they will be able to do the task better. So there's a difference between perception and action in these cases. That brings me to the summary of the second part. And I see that there are a couple of questions in this. One of them is very long, so I'm going to just maybe quickly just summarize the third part as well and spend some time on the questions. So the third part is actually got to do with the AI ML model that we have sort of developed for the keratoconics detection. Sorry, before I do that, just a quick summary of what we did. Very complex pattern of blur in keratoconus, leading to very complex vision loss. The tools that we need or we have used for addressing these problems certainly go beyond routine optometry techniques, and I have listed out some of these tools. And the vision loss seems to be about 80% related to optics, but there seems to be a 20% of loss coming from the neurology as well. So we need to start involving the brain also in our thought process when we are dealing with keratoconus. We know very little about the oculomotor status of these patients, how stably they fixate, what is their accommodative responses, Virgin's responses. We know very little about that. These are all some of the investigations that are currently ongoing in the lab. And if Gopi and Saranya invite me once more in a year's time, I'm very happy to talk about those, hopefully. The last part, very quickly, prevalence and incidence of keratoconus is very sparse across the globe. In this picture here, India seems to have a very high prevalence rate, and that is only because there is a single data point that is also from adults. So we really do not have any idea of prevalence or incidence. One of the challenges for this is present diagnostics is all these super fancy techniques. These are all very good at diagnosing it, but you can't carry them into a mass screening endeavor. These are too expensive, non-portable, and all of that. So what we did was we actually explored the possibility of using photorefraction as a technique for measuring keratoconus. This is a gold standard technique available for measurement of routine refractive errors. Here are some commercial devices that are being used. So we have our own version of the photorefractor that we used to measure these profiles. And what we found was the pattern of pattern of profile in keratoconic eyes is very different from that of controls. There is a signature that is here for us to actually detect keratoconus. And using that, I worked with some of my colleagues at IIT Hyderabad. and we built an AI and ML model for us to see if these images can actually be used as a good diagnostic, good classification for keratoconic images. Turns out we had, so there are 3-4 different segments in that model, but in the interest of time, I won't get into the details. What we found was, good accuracy of almost 86%, and we had some really interesting geometric features that this model developed, which we are currently using now to build on this model, build on this AIML model. So, using artificial intelligence, machine learning techniques, you can use them as early detection markers for keratoconus as well. So with that, I'm going to, so this is what I spoke about. With that, I'm going to conclude saying we covered a whole range of different topics related to keratoconus. Most of it was actually spent on looking at the structure function relationship, how complex the structural loss is, what's the consequences of this for form vision and depth vision. We also then discriminated between performance at a threshold level, acuity, contrast, stereo, versus performance at a supra-threshold level, relating to more real-life experience. And in the last part, we also spoke about more efficient detection of keratoconus using a presently used gold standard technique for refractive error screening, photorefraction techniques. So, in summary, the disease is a very complex one, and you need complex techniques that are outside of the optometry domain to actually be able to solve this. So, collaborative work with other domains, engineers, mathematicians, psychologists, I think, mandatory at some level in order for us to address problems like this. This is not unique to keratoconus. Almost any complex disorder, you may want to go outside of your comfort zone and your domain and look at all of these other areas and bring in those kinds of resources. So with that, I will stop. And thank you very much for your time. Gopi, Saranya, thank you so much for the invitation for this. I'm happy to take any questions.
Thank you so much, sir. So we have good number of questions.
Tons of questions actually, Shrikant. I've just been
Okay.
reading.
I'm
Gopi,
seeing
you want
3
to
of
do
them
the...
here.
Yeah, the
Or
first
maybe,
one
oh,
is
no, there is a whole range of them. Sorry. Okay.
No, there's a whole load. So the
Okay.
first one is, even after giving rose scale lenses to one of my patients, she's still not getting pinpoint images. What is the
Yeah.
next solution?
So one, see, I'm not full-fledged in the clinic for me to be offering clinical solutions, but my suggestion will be that I would probably try a scleral lens. The reason why it is not working is because the aberration patterns are not fully corrected by the rose scale lenses. So I would probably try out a scleral lens, probably a better fit for the scleral lens that might help us help your patient, sir. Would advising patients to tell contrast.
So the next question was, would advising patients with KC to increase environmental contrast be a beneficial strategy for enhancing the visual function?
I think it is, Anusia. It will be because what you're doing by enhancing the contrast is you're going from a threshold level performance to a supra threshold level performance. So you're giving more energy to the keratoconic system to actually do its task. We as normal adults, if we benefit say 10% from that improvement in contrast, the keratoconic system is going to benefit a whole lot from it. So I think that's a good idea. What higher illumination will also help, the reason it will also help is because it will produce pupil meiosis, and the minute pupil meiosis starts happening, your wavefront aberrations reduces. That will also improve the visual performance in these keratoconic eyes.
So Swapnil has a long question for you.
Yeah. Adaptation to multifocal optics. Swapnil, I don't have a direct answer for you, Swapnil. There is circumstantial evidence that if you have a multifocal optics, if you get exposed to a multifocal optics for extended periods of time, say an hour hour and a half and stuff, certainly visual performance improves in these patients. And the reverse is also true. If you are now sort of living with multifocal optics for very, very long periods of time, then there is a certain amount of amblyopia that is actually kicking in. So it's a trade-off between a short-term period where your visual system does try and adapt it, but if that now persists for a long period of time, the strategy used by the system seems to be different. If you are asking, okay, what is exactly changing in this? I don't have an answer. I don't know that anybody has an answer for this question. Neural adaptation as a term is one of the most misused terminologies. Anything that you cannot explain, you put the blame on neural adaptation. So I'm going to continue to do that and saying this is some form of neural adaptation. I don't know the answer more than that.
So, Shrikant, just for people who don't understand the term multifocal optics and keratoconic eyes, could you just quickly say what it is?
Yeah. No, I can do that. So when I say multifocal optics, I am comparing that to a monofocal system. So all the light rays entering into the eye comes to a focus at a single point. Now that single point could be a myopic point or a hyperopic point or emmetropic, but nevertheless, all the light rays are coming to a focus at a single point. Multifocal optics is light rays entering the pupil from different portions of the eye come to a focus at multiple different points. Spherical aberration, for instance, is a good example of a multifocal optics. Light rays going through the center of the pupil will focus somewhere, through the periphery of the pupil will focus somewhere else. So that's so keratoconic system, because of the aberrations in the system, it is inherently a multifocal optical system.
So, I mean, going with this, would you say correcting them as early as possible would, say, a contact lens be the better idea?
I think so, Sharanya. It's a better idea for two reasons. One is the optical quality of the eye. You are actually presenting, good quality of the optics very early on in their disease condition. Secondly, if you are considering bilateral corrections, you are actually making it similar also in the two eyes. Right. So one of the things for me that's fascinating about keratoconus is by the time they, in the present state, you are only able to diagnose keratoconus for patients who are coming to the clinic, which means they are already sort of experiencing some form of degraded vision, which is why they come to you. When did the insult happen? Could have happened several years earlier. Now, from that point on, these are individuals living with blurred vision for extended period. All our understanding of amblyopia will tell you that these patients should have become amblyopic at some level or the other. So, how do we overcome that is fundamentally the question. Did clinical stereo match the visuomotor measurements? So if you are following my talk, RD, turned out it did not match. And the reason being is that our clinical stereo measurement is a measure of perception of depth. You're not asking anybody to go do a motor action. Motor action is more complex because once you perceive the depth, you need to move your hand to the specific location and move around and things like that. That's a lot more complex task, which is why I feel that perception does not necessarily match the motor actions. Right. Without stereo acuity, individuals also have monocular cues to depth. Absolutely. Yeah. Will keratoconic eyes? This is the great question by Terry. So, before we got into the keratoconic world, we actually explored this in individuals with only one eye. Habitually one-eyed individuals who are now having to depend only on monocular cues to depth. Are they able to do it? In the visuomotor task, they were not able to do it. They were not able to do the monocular cues. The reason is the task is a very complex task. If I go back to the that picture, and it will become hopefully a little bit more obvious. Where am I? Yeah, so there. In that picture, you see the task of the person is to constantly take this hoop and they will have to move this in and out. Now, one of the monocular tasks that monocular cues is motion parallax. So you can move like a bird and then try and get depth information. Now imagine doing this task. You're also moving this. You're also moving your head around. You're also having to look at other kinds of clues. It becomes a total mess in the visual system. So what Preeti is currently doing is she's recreated a static version of this task. You simply look at it and say, okay, is that in contact or not in contact? And the one-eyed individuals are really good at it. So they are able to use it. They are able to use it for perception, but not for motor action. We haven't done the equivalent for keratoconus yet. Hopefully, with Preeti finishing up her thesis, and we'll have to see how we can move it forward. But that's certainly on the anvil for us to do that experiment. Will stereo blind use contiguous temporal disparities as cues of depth? Yeah, no, absolutely. This is the motion parallax that I was talking about. So contiguous temporal disparities is essentially I sample image in one position, I move my head over and I sample image in the other position. I now compare what was the depth between time here and time there, which is essentially your motion parallax signal that you are using. Like I was saying earlier, motion parallax seems to be a good cue for perception of depth, but for motor action, at least for a task like this, it seems to be fairly useless in these one-eyed individuals. Right, okay. Multifocal lenses have hard and soft designs. Soft designs only. Ravi Shankar sir, I am not entirely sure I have an answer for this. Maybe Sharanya, you are more of a serious contact lens practitioner that I am.
No, I think he's talking about progressive addition lenses and glasses.
I see. Okay, I see. Yeah. Okay, is there Augustine is asking, is there a Zernike coefficients can be transformed into optical assistive devices? Would they be effective as a solutions for aberrations? Yeah, most certainly. Most certainly. Now, these days, you actually have wavefront corrected contact lenses for keratoconus. So the measure of the wavefront aberrations is the wavefront aberrations of the patient is measured. The mirror image of that is actually printed on your contact lens surface. And when you wear one of these, you are now going to essentially cancel out these aberrations. There are commercial designs that are being launched. There are experimental designs that are available. The one challenge with these customized contact lenses is the fit of these lenses in the eye. Because even if these lenses move a little bit in the eye, now you have a whole different set of aberration patterns, which will create a hell for you. So you want to sort of be careful with that, but certainly that's where people are thinking about it. What's a possible explanation? Specialty CLs came same as RGP. I think the explanation is in the quality of the retinal image side. The through focus curves that I was showing you earlier had very similar shapes for RGPs and your specialty lenses. What this means is that any of these lenses are able to correct your wavefront aberrations only to a certain extent. There is a certain residual that is always left in these with these lenses. It's these residuals that is actually limiting performance. And what this tells us is the amount of residuals in these wavefront aberrations is similar across these different designs. All right. So yes, I think
I think you covered all the questions.
Yeah.
Okay. Very nice.
Well, very interesting that people were able to. So a few years back, I didn't think people would be able to understand and take it in and ask you questions.
Yeah.
So I'm actually quite happy that either those who are serious are really getting more serious and listening and learning.
Absolutely.
And I'm really, really happy to see that.
Yeah, I am impressed. I am very, very impressed. I thought this will be very esoteric, whatever I am talking, but no,
So the thing is, Shrikant, we've been very surprised with the response that we get with people on the Inspire sessions. This
Very
is an
nice.
open
Yeah.
session that we actually planned because we wanted to introduce people to the opportunities that are available for optometrists. It's not just refraction, and I'm not just a contact lens specialist, or I'm not
Correct.
just into BD. There's so many things that you can do, and the more you do and the more you specialize and the more you explore the options, you are going to find yourself in a better position career wise. That holds
Absolutely.
across the board on any field. It's
Absolutely.
just that with optometry, we've never had an option to showcase all these things, and this is what we really wanted to bring out to any optometrist who's out there. So this is a
Correct.
free
Correct.
session which is open to everybody. We post it out in almost any and every group which is available, and we get a lot of good responses. We actually put it on our YouTube channel, which is available for viewing later. So we've
Right.
actually had people view those and come back and ask us questions as well.
Very nice. Yeah.
So,
No, if there is
so
anything like this in the future, I mean, in the YouTube questions, I'm happy to answer those as well.
thank you, thank you. We really, really appreciate that.
Yeah. And for
And
those of you,
yeah, and
no,
I really have
before
to
you
thank
leave, I have to
Gopi
say
for
this. For
no,
those
no, no.
of
I
you who
really have
don't,
to thank Gopi for doing all the background work on all this.
yes.
So I just tell him, can you get these people, and I just leave it to him, and he
Yeah.
does
Yeah.
all the
Absolutely.
follow up and everything. So thanks, Gopi.
Remarkable, remarkable,
Thank you.
remarkable. Yeah, one thing, Gopi, before you end, I want to say this for the audience that are left behind. Sharanya was my teacher in optometry school, and she has been a source of inspiration for me all along. So whatever she
Oh,
has said
thank you.
about overall development of the profession, I can't agree with her more, 200% more. So for those of you who are in different stages of your careers, please do consider what she's saying very, very seriously. This lady has a lot of weight to her. Thank you, Saranya.
Thank you so much. You don't know Shrikant. When I actually went to UCB to meet Pia, I initially didn't get an appointment to see her. But then I sent her a letter saying that I'm from the Elite School of Optometry and like to observe her lab, the BB lab.
Yeah.
Apparently, she had a chat with you, and you mentioned, I mean, some email correspondence, and you
Oleg,
mentioned,
is it?
she
Yeah,
said,
Pia Heinig, yeah.
yeah. So she said, yeah, please, she's anything. So thank you.
Sure, sure. My pleasure. My pleasure.
Yeah, thank you so much.
Thank you, Dr.
Yeah.
Shrikant, and all the listeners for the good questions. And we will again post it in the YouTube channel, maybe in a couple of days. If you want to rewatch it and then connect with Dr. Shrikant, maybe I will add the email ID as well in the description of the YouTube link, and you can connect with him, and maybe we can. So, and overall, I believe collaboration with multidisciplinary is going to be a key future research. And thank you again for joining. Thank you, everybody.
Thanks, Shrikant.
Thank you. Thank you so much.
Thanks, Sophie.
Again, my apologies, guys. So where I was